The overall goal of this procedure is to grow Pseudomonas aeruginosa biofilms at the apical surface of live human airway epithelial cells. This is accomplished by first establishing a confluent monolayer of airway epithelial cells on a plastic surface or a glass cover slip. The second step of the procedure is to grow a culture of P. aeruginosa bacteria constitutively expressing the green fluorescent protein. The next step is to place the bacteria and airway cells in contact with each other in either a static setting or in a flow chamber. The final step of the procedure is to let bacterial biofilms develop over time while assessing the viability of the airway cells. Results can be obtained that show biofilm formation on live airway cells through fluorescence microscopy.
The implication of this technique extends toward therapy of cystic fibrosis patients because these co-culture models can be used to develop and validate new antibiotic regimens able to eradicate biofilms responsible for chronic colonization of the airways in cystic fibrosis patients.
The static co-culture biofilm model uses CFBE cells, which are immortalized human airway epithelial cells originally developed from an individual with cystic fibrosis. 7 to 10 days prior to bacterial inoculation, seed the CFBE cells in a 24-well tissue culture plate. We seed cells at a concentration of 2 × 10⁵ cells per well in 0.5 milliliters of minimal essential medium, or MEM, supplemented with 10% fetal bovine serum, 2 millimolar L-glutamine, 50 units per milliliter penicillin, and 50 micrograms per milliliter streptomycin. Grow the cells at 37 degrees Celsius and 5% carbon.